Hello, today I'm going to discuss about the topic glycolysis. The glycolysis is the common pathway to both aerobic and anaerobic modes of respiration. The common pathway of aerobic respiration include glycolysis, the Krebs cycle and TCA cycle. TCA cycle or ETS cycle, it is also known as ETS cycle, electron transport chain. So now here we are going to discuss about the glycolysis. What's glycolysis? Glycose means sugar and lysis means splitting. So the glycolysis means the splitting of the sugar. The glycolysis is the partial oxidation of the glucose into two molecules of pyruvic acid. That means the glucose is converted into two molecules of pyruvate. So this occurs in the cytoplasm. Now the question arises how the glycolysis can be regulated. The glycolysis can be regulated by the amount of glucose that is available for the process of the glycolysis. The amount of glucose that is available for the process of the glycolysis. The glucose is available for the process which is regulated primarily by the two ways. Now the glucose is available by two ways to our body, that is by the regulation of glucose reuptake and the another is the regulation of breakdown of glycogen. Breakdown of glycogen. The glucose transporters. They are the glucose transporter that transport the glucose from outside cell to inside. So the glucose transporters, they are of 5 types. Glucose transporter 1 that are found in RBC, the glucose transporter 2 that are found in liver, in kidney, in gastrointestinal tract, and in beta cells of the pancreas. The glucose transporters 3 are found in neurons. The 4 are found in heart and in skeletal muscles. Whereas the glucose transporters 5 are found that transport specially fructose into the cell. The main function is to transport the fructose into the cells. And another way is regulation of breakdown of glycogen. So the excess of the glucose that is in the body, that is stored in the form of the glycogen in the liver. So when the amount of the level of the glucose decreased in the body, then the glycogen starts breakdown into the glucose. Now the glycolysis pathway. The glucose is converted into glucose 6-phosphate with the help of enzyme hexokinase or glucokinase. One thing to keep in mind that when the either ATP is converted into ADP or ADP is converted into ATP, the enzyme kinase is used. This is the easier way to remember about the enzyme. So after the conversion of the glucose 6-phosphate, The After the conversion of glucose into the glucose 6 phosphate, the fructose 6 phosphate is formed. The glucose 6 phosphate is converted into the fructose 6 phosphate with the help of enzyme isomerase. Isomer are that the change in position. Now the fructose 6 phosphate that is converted into the fructose 1,6 bisphosphate. Now one phosphorus is come from the ATP. In the presence of magnesium ion, the enzyme phosphofructokinase is used here. So the fructose 6 phosphate is converted into fructose 1,6 bisphosphate. So now with the help of the enzyme aldolase, the 6 carbon molecule is converted into two 3 carbon molecules. These two are the 3 carbon molecules that are dihydroxyacetone 3 phosphate and glyceraldehyde 3 phosphate. Both are the 3 carbon molecule and contain the phosphorus group or phosphate group at the third carbon. The dihydroxyacetone 3 phosphate DHAP that is interconverted into glyceraldehyde 3 phosphate with the help of enzyme triose phosphate isomerase. So now these both are in the equilibrium. Now the glyceraldehyde 3 phosphate is converted into the two molecules of 1,3 bisphosphoglycerate with the help of enzyme dehydrogenase. Now where the dehydrogenase enzyme comes, the NAD positive is converted into the NADH. So now 1,3 bisphosphoglycerate is converted into two molecules of phosphoglycerate. The kinase enzyme phosphoglycerate kinase enzyme comes which converts the ADP into ATP. So keep this in mind here, the ADP is converted into ATP and in the above steps the ATP is converted into the ADP. So now the phosphorus that is present on the carbon first, that is removed with the help of the enzyme kinase, and now the remaining left is 3-phosphoglycerate. And now the 3-phosphoglycerate undergo the mutation and changes into the 2-phosphoglycerate with the help of enzyme mutase. As it undergoes the mutation, so the mutase enzyme is used to convert the 3-phosphoglycerate into 2-phosphoglycerate. Now the enolase enzyme comes which release the water and that converts the 2-phosphoglycerate into 2-phosphoenol pyruvate. This is PEP. Now the two molecules of phosphoenol pyruvate are formed. And the last enzyme comes is the pyruvate kinase. Here also the ADP is converted into ATP. So in the last step the two molecules of pyruvate are formed. So now the glycolysis pathway can be separated into the two phases. The first phase is the preparatory or the investment phase, and the second phase is the payoff phase. The investment phase or the preparatory phase are that phase wherein the ATP is consumed. Like here, the ATP is consumed. Here, ATP is consumed. The ATP is converted into ADP. One molecule of phosphorus is consumed. Here also the one molecule of phosphorus is consumed. And the another phase is the payoff phase, where the one molecule is produced, one ATP is produced, that is the phosphorus from 1,3 bisphosphate from the first carbon that joins the ADP and releases the ATP. Here ADP is converted into ATP, and here also in the conversion of phosphoenol pyruvate to pyruvate, the ADP is converted into ATP. Now, in the complete glycolysis pathway, the two molecules of ATP are consumed and in turn, 4 molecules of ATP are produced. Here, the two molecules, first molecule of ATP is consumed and the second molecule of ATP is consumed. And in turn, the two molecules um, why they are the two molecules? Because the two molecules of bisphosphoglycerate is converted into two molecules of 3-phosphoglycerate. So here the two molecules of ADP are produced. The two molecules of ATP are produced, and here also the two molecules of ATP are produced, and in turn the four molecules of ATP are produced. Now the NADPH, each NADH is equivalent to each molecule of NADH, that is equivalent to the three molecules of ATP. So net how much ATP in the glycolysis are present, or net gain in the glycolysis of ATP is 2, 4, 2, 4, 6. So the net gain in the glycolysis is the 8 AP molecules. Thank you, keep studying, and all the best.